Okay, let's try this. So we were talking about recombinant DNA technologies and how we could cut and paste DNA in novel combinations, and the question is why bother to do any of that? And the answer is partly because you can make fusions between genes and other genes in novel combinations. So an example we went through before, this is reviewing, is if you want to make a large amount of a certain protein, you can fuse the gene for that protein to a very strong promoter. And we went through the T7 technology in some detail. So then we went to another useful kind of fusion would be to fuse if you want to study a particular promoter, you fuse that promoter to a gene whose product is easy to measure. And what is widely done in hundreds of laboratories is to fuse promoters of interest to our old friend LAC-Z. LAC-Z is easy to measure because a large number of artificial substrates have been created that, when cleaved, turn color, or they become fluorescent or luminescent or what have you. And so that makes the enzyme very easy to quantitate. And so that is widely done. An example that I think we might have touched on is this. We remember the Rccombinant system. Lexa represses many genes, and it is cut in half by, with assistance from Rcca when there is DNA damage. You remember all that. So if you take one of those promoters and fuse it to LAC-Z, then when the cell experiences non-lethal amounts of DNA damage, it responds by synthesizing beta-glycosidase. So that's easy to quantitate. So then you can ask, how much UV, what wavelength UV, what other kinds of DNA damaging agents, what other kinds of stresses, what if you mutate Rcca or you mutate Lexa, what happens then? All that can be tested by measuring the production or lack of production of beta-glycosidase. Okay, so that's done in hundreds of labs. Instead of using LAC-Z, there are other types of genes that are easy to measure. These genes in general are called reporter genes. They report on the expression of a promoter. Another widely used reporter gene is called GFP, green fluorescent protein. It originally comes from a jellyfish. It is a protein that can absorb blue light and fluoresce green, which may sound so what, but in fact it's very easy to take a microscope and outfit that microscope to excite the image with blue light and then filter out everything but green light, and you can detect and quantitate the green light that is released. That's used in many labs. You can also fuse these genes to make fused proteins, and the fused proteins can then be localized in the microscope. This is hugely important technology. The GFP has been modified to fluoresce at different colors. So just focusing at the bottom there are variants that absorb green light and fluoresce red, for example. So you can have 2 or maybe even 3 different fluorescent proteins in the same experiment, and they can report on different promoters or different. So that's widely used. This has also been commercialized. It's possible to go out and purchase fluorescent aquarium fish, fluorescent puppy dog. I don't know why that was done. I hope it wasn't just to be clever, but it's out there. You can do fluorescent artwork. And so, okay. Another example of an interesting and useful fusion is the following. I just realized I have no cursor. Let me fix that. Cursor visible. Okay. Now I have a cursor. All right. What is possible to do is to fuse a protein of interest. It'll go over here in this little, up in the upper right hand corner is the protein of interest, and at the left end, the N terminus has 6 histidine residues. Okay, so this is a fused protein. It is the full protein that you want to study, but at its N terminus there are 6 extra histidines. It turns out that makes this protein, this fusion protein, is extremely easy to purify. One can purchase beads, microbeads. These are micro. Go down to the bottom of the slide, microbead. This green line here is meant to be the surface of a bead that is very tiny, maybe 50 microns. They purchase these beads, and they have on their surface, and actually inside as well, but let's imagine the surface here. Here is a chemical that tightly binds nickel atoms, single atoms of nickel cations. And you purchase these beads, and the beads look kind of blue because they're so full of nickel. And you use those beads to make a column. This is a cylindrical column full of beads, and you can add proteins to the top, and they wash out the bottom. So you have an E. coli strain that makes large amounts of a fusion protein consisting of the protein of interest fused to these 6 histidine residues. In some cases, the histidines will interfere with the function of the protein, but in most cases they don't. And so you can add, here is a lysate of the protein of interest and all the other contaminating proteins that you'd like to remove. And so you put the whole mixture onto the top of this column. The protein of interest binds tightly to the nickel atoms that are immobilized on these beads. And there's showing 2 histidine residues on the long polypeptide of this protein. They're binding tightly, and the entire protein is stuck to the nickel atoms, and the nickel atoms are stuck to the beads. So, and all the other proteins hopefully don't bind, and they are released. They go right through the column out the other end, and they are discarded. So when the column is sufficiently clean of all the other contaminating proteins, now you have to remove the protein of interest. You elute the protein of interest. It is done by using a molecule that resembles histidine. The molecule that we use is called imidazole. It's over in the lower left. You probably could use histidine itself, but we never do. We always add imidazole, and that competes against the histidines because it likes to bind. You can see they're pretty much the same molecule, and it will also like to bind to the nickel. And if you add enough of it, it will outcompete the protein, and the protein will then be released. So it's a pretty simple process. If you have the column, and you have the protein of interest fused to histidine residues, you can add it to the top of the column, have all the contaminating proteins wash out, and then add some imidazole and have your protein elute. Very pure in a very small volume. It's a lovely technology. Now, how do you make this fusion protein? Well, over in this toward the upper part of the middle of the slide here, there is a plasmid that you can obtain, and you can cut that plasmid with restriction endonucleases and insert your gene of interest here. But this plasmid has a strong promoter. It has 2 copies of the LAC repressor binding site. It has it encodes a ribosome binding site, and it has encodes an AUG start codon. And then in the same reading frame, after the ATG, are 6 codons that encode histidine. And then you put in the gene of interest, and you have to make sure that the gene you're inserting is in the same reading frame, the same register as the ATG. Because when this is converted to RNA, and the ribosome binds here, and the AUG establishes the reading frame, it will read the histidines in the correct reading frame and make the histidine codon, insert the histidine amino acids. And then you have to make sure that your gene is in that same triplet reading frame so that it will make the ribosome will synthesize a fusion protein. And that's how you do. You insert your fragment into one of these plasmids and make the fusion protein, and it will then make that protein. And it's a beautiful way to purify a protein in high yield very quickly. Okay, those are 3 examples of how one could make fusions between novel combinations of genes for obvious purposes. Okay, let's move on to how does one sequence DNA? How does one obtain gene sequences? What I'm going to describe is one of many technologies. It is becoming a venerable technology, but still widely used. It involves using specialized substrates for making DNA called dideoxy NTPs. So in ordinary, this on the left we have an ordinary DNA base, a deoxy NTP. The N means any base. Here's the deoxyribose, missing an OH at the 2 carbon. It has the OH at the 3 carbon, and it has at the 5 carbon the triphosphates. Okay, so you've seen that before. This is very similar. This is a dideoxyribose. It's pretty much the same, except that it is missing an OH at that position. It is a deoxy at the 2' position and the 3' position. Okay, that is a critical idea. So these bases can be easily incorporated into a growing chain because they're normal at the 5' position. But so here it is. Here's the base that has been just incorporated into the growing chain of DNA, but the deoxy makes it impossible for that chain to grow any further. So we call that a chain terminating substrate. It terminates chain elongation. Why is that useful? Well, what's been done, this is kind of a busy slide, but let's go through it. Here we have the 4 bases. Okay, so here we have a thymine, and here we have a dideoxy, DDT. Over here is the ordinary triphosphate. It's written a little differently, but it's the triphosphate. So that by itself is a deoxy T. I'm sorry, it's a dideoxy T. Here is a dideoxy C, a dideoxy A, a dideoxy G. But what's also here is this great big molecule, which is a fluorescent molecule. And there's 4 different ones. If you look carefully, you'll see that this is not the same molecule as that. And these are all 4 different. They all absorb light and they fluoresce at different frequencies. Okay, so we purchase dideoxy T with this particular dye, and you purchase dideoxy C with this tag, and dideoxy A with this fluorescent tag, and this dideoxy G with this fluorescent tag. Okay, so and then what one does is to polymerize DNA with them. Now let's look on the upper left. You can see we're going to have a mixture. We're going to have a DNA template and a primer, which is shown better on the next slide. But here we focus on how we have the 4 ordinary substrates, deoxy A, G, C, T, and limiting amounts of fluorescently labeled dideoxy A, dideoxy G, dideoxy C, dideoxy T, and of course these colors indicate that these are fluorescently labeled with different fluorescent tags. Now, I think maybe the next slide shows a clearer picture of how they're used. Let's go there. Here we have something I drew. Here we have a DNA template, and this would actually be obviously much longer, but just to show it, keep it simple. Here we have an oligonucleotide that is going to serve as a DNA primer. So synthesis of DNA will occur at this A, from left to right. Okay, in the first base is going to be opposite of C, so it has to be a G. G will be inserted here. And we have a large excess of ordinary G. So 99% incorporate an ordinary G, and they are not fluorescent, and they can be further extended. So here we have the ordinary G inserted. There's no fluorescent tag, and that can be extended further. But 1%, I mean the number 1% is kind of a made-up number, but imagine 1% of these templates incorporate a dideoxy G that is fluorescent. So those molecules, that 1% of these molecules, are now they can no longer be extended, and they are fluorescent. And they are 8 bases. This is 8 bases long. Okay. Now the next base to be incorporated is a C opposite this G. And there it is. 99% incorporate an ordinary C. They're not fluorescent. They can be extended further. 1% incorporate a dideoxy C. They cannot be extended further. They have the dideoxy. They cannot be extended, and they are fluorescent. And they are fluorescent red. And they are 9 bases long. So now we have a population of molecules that are 8 bases long and they fluoresce green, and a population of molecules 9 bases long that fluoresce red. And most of the molecules don't. They don't have any fluorescence. because they're going to extend a third and a fourth and a fifth and a sixth and a seventh round of synthesis. And each step, a small fraction will be terminated and fluorescent. The rest will continue on. So you end up with, going back a slide, you end up with this. You end up with a series of molecules of different lengths, and they're fluorescently labeled with different colored dyes. Uh-oh, I have to pause now.